Mainly talking about vocabulary and just a basic background to heredity and genetics. So, what vocabulary is important for understanding genetics? Well, first off, we need to know gene, since that word is literally in the word genetics. Now, what a gene is a small section of DNA that codes for proteins. Remember, in previous lectures, we talked about transcription, translation, making those proteins, and your gene is basically that instruction. An allele, on the other hand, is an alternative form of the gene, a variation of the gene. That arises by mutation and found on the same chromosome at the same place. So this is when we get into, remember, homologous chromosomes. You get one chromosome from mom, one chromosome from dad. And they are going to have similar sizes to them, as well as similar genes. We call them loci, so the gene locations. In this case, we have this chromosome has the gene for purple flowers, and this chromosome has the gene for white flowers. So our overall gene is flower color, and we have two different alleles, two different forms of the flower color gene. You have purple and you have white. So we get variation from mutation. Now, genome is simply all of the genetic material of an organism. We have successfully mapped out the human genome, so the ability to potentially clone a human is becoming more real with each passing year. They did just successfully clone I believe a monkey within the past year. Your genotype is the complete heritable genetic identity. So a pretty official definition, your genotypes, I'm sure you've heard the words homozygous dominant, heterozygous, homozygous recessive. So we basically only have three types of your genotype. Now with our symbols, if you see a capital letter, we call that a dominant allele. We have letters to basically represent those gene sequences. Instead of writing the sequences over and over and over, we symbolize them by letters. So a capital letter. So for instance, if I just have like a capital A, and then a lowercase letter we call recessive. And a lot of times it's easier to have letters that have a completely different capital version versus a lowercase version, just to make it easier to reference. Now, genotype and phenotype are connected. So we'll go to that next. Your phenotype is the observable, and that's a key word here, observable physical characteristics of an organism. Whoops. So these are going to be the physical traits, and these are going to be coded by the genotype. So if we look down here at the picture, your genotype, let's say, might be, BB, based off of, these two genes right here. Well, let's say two capital B's codes for a blue color. So the genotype is going to be those two allele combinations. And then the phenotype is the physical trait that it is coded for. Coming back up here, we do have a couple different types of genotypes as stated before. So we're going to throw out the words homozygous. What homozygous means is we use the word purebred. So this is going to mean basically identical alleles on the homologous chromosomes. So you basically have the two extremes. So what I mean by that is, if we go back to our letters right here, this is going to be either two capital letters or it's going to be two lowercase letters, two dominant letters or two dominant alleles, two recessive alleles. So that's what we call purebred, having the extreme. One extreme or the other. On the other hand, we also have heterozygous, where we have two different alleles on the homologous chromosomes. And we would call this a hybrid. And this is when we have one of each. One capital, one lowercase. So two out of our three situations, we call it a purebred. One out of the three, we call it a hybrid. Now, when we talk about our dominant and recessive, what we're getting into is dominant is only expressed when one copy is present. So we would say that there is a dominant phenotype whenever we have two capital letters or one of each. The fact that we have one capital letter, the dominant phenotype is going to show. Whereas recessive, this is only expressed whenever both of your alleles are lowercase. So two lowercase letters. We call that homozygous recessive. So a little bit of vocabulary. In terms of understanding some of the terminology we're going to be discussing over the course of the next probably 2 weeks. Now, in terms of how this is all connected, is we're going to talk about Mendelian genetics. And if there's one thing to understand about Mendelian genetics, is we are assuming what's called simple dominance, where what this means is these two will show the dominant trait. And then this one will show the recessive trait. So that's what we are assuming when we call simple dominance. Now this all stems back from an Austrian monk called Gregor Mendel. Did a lot of studies with what he's famously known for with his pea plants. Pea plants are very basic, very observable characteristics, very easy to fertilize as well as observe, pretty fast growing patterns as well. So in tying in genetics with what we just covered with meiosis, we kind of get into ideas of probabilities and predictions of what could happen. And Gregor Mendel's experiments are all based off of that simple dominance. 2 out of our 3 genotypes will code for the dominant allele. 1 out of the 3 will code for the recessive allele. Now he followed 3 different laws of inheritance. We have briefly touched upon the law of independent assortment, and then we're going to talk about the law of segregation, which kind of goes with it, with meiosis as well, and then the law of dominance, which is what all of his experiments are based off of. So when we look at the law of segregation, remember, we are going from a diploid to a haploid cell. So this comes back to meiosis. Where meiosis is going from diploid, remember we have 2 copies of everything, to haploid, where we have 1 copy of those chromosomes. So the key thing here is understanding that if we're talking about your father's sperm cells could carry the X or the Y. And depending on which sperm cell fertilizes the egg cell, that's going to help determine what your gender is. And that's kind of how the law of segregation works, is each parent is only providing 1 allele. They have their own combination of alleles, which gives them their phenotype, as well as genotype, but they can only pass 1 of them on. So this leads to potential having options in terms of probability. What's the chances of your parents giving you this or this? Now, the law of independent assortment. Remember, in meiosis, once again, we are talking during metaphase 1. During metaphase 1, we have options in terms of how those homologous pairs line up. They can kind of mix and match. Not looking at this first circle right here. Let's say the purple is your mom's chromosomes and the red is your dad's chromosomes. Your mom's chromosomes don't necessarily all have to line up on the left and your dad's on the right. Now just imagine there is a line of 23 pairs of chromosomes. They can kind of mix and match, and that leads to a lot of variation possibilities in the end. So the more chromosomes you have, the more possibilities you have of variation. So when you look at more and more and more variables, your Punnett square is going to get bigger and bigger and bigger. And then finally we have our simple law of dominance. Now, Mendel is very famous for his studies with his pea plants. What he did is he took what's called the parent generation, took purple flower and bred it to a white flower and ended up with all purple flowers, which brought in the idea that, purple must be dominant over white since we've got all purple. So he started through a lot of different experiments, find that you only needed 1 purple allele to show that purple color, whereas you needed 2 white alleles to show the white color. And how he found that out is he ended up self-fertilizing what's called the F1 generation to create the F2 generation, where the white kind of popped up again. So what this is telling us is that in the initial generation, we'll use F just to make it easier. We had 2 purebred plants, and we ended up with all hybrid offspring. Now, just for kicks, what he ended up doing was self-fertilizing. So we basically have a hybrid cross here where we end up with 3/4 purple, 3/4 dominant allele, and then 1/4 homozygous recessive, 1/4 recessive allele being shown. So 3/4 purple, 1/4 white after showing that hybrid cross. So just understanding that purple was dominant over white. I used F for that and lowercase f for here. And we see that through 2 different generations of crosses. All this can kind of be traced back to meiosis with your segregation and your independent assortment leading to options. This is simply probability. So there's still chance in terms of what could end up happening. And what we're going to end up with at the end of the week is starting to calculate this probability. And that's when we get into what's called your Punnett squared. We put one of the parents across the top. We put one of the parents going down vertically. Whenever you are only dealing with one trait, we call it monohybrid, mono meaning one. So we basically have 4 possibilities that could happen. So 4 squares. Whenever you are getting into 2 or more traits, your Punnett square gets bigger and bigger. So 2 traits, that is now leading to 16 squares. Same thing, we have 1 parent across the top, 1 parent across the bottom. And as you get bigger and bigger and bigger in terms of more variables with traits, your Punnett square is going to get bigger and bigger and bigger as well. And a lot more options in terms of what's happening. Over here, we only have 2 options. We basically have dominant recessive for 1 trait. Over here, we have 4 options. We have dominant dominant. You have dominant recessive, recessive dominant, recessive recessive when you're looking at 2 traits. And we'll get to this later on. Just kind of wanted to show you a visual in terms of the differences. The reason why we have more squares with a dihybrid is due to that law of independent assortment where we could have options. So more options in terms of what's happening. All right, that is it for our intro today. If you have any questions, please ask. Thanks for watching.